Now I will talk about the role of oxytocin neurons in non-reproductive behavior. Oxytocin neurons have been linked to a variety of parental and reproductive-based social interactions and behaviors, such as those we have previously covered in this resource. Yet, less is known about how oxytocin affects non-reproductive behavior and how it influences same-sex social connections. A great mammalian model to examine the neurocircuitry involved are female meadow voles as a result of them living in social nesting groups during winter months. Beery and Zucker identified that the lateral septum and the central amygdala, which are not known to be associated with opposite sex affiliation, showed a connection between oxytocin receptor density and social behavior. This research demonstrated that oxytocin functions differently from neurocircuits linked to sexual bonding. Rat social systems are much simpler than those of primates. There are many more opportunities and techniques to examine how oxytocin influences non-reproductive behavior in humans and primates. Schweiger et al. Showed how oxytocin influences some of these non-reproductive behaviors. They demonstrated that oxytocin increases one's ability to infer emotion. Their research reported that intranasal administration of oxytocin within adults who experience adverse childhood experiences resulted in an increase in score of the reading the mind in the eyes test. Oxytocin has been shown to substantially increase trust within humans. This was demonstrated by Gosfeld et al., and their work showed that intranasal administration of oxytocin specifically impacted one's willingness to take on social risks brought on by relational encounters. As previously mentioned, research on trust has shown that oxytocin interacts with the amygdala and influences how emotions are processed. Studies evaluating the impact of oxytocin on specific emotions were inspired by this connection. This relationship was observed by Kirsch et al., who employed mRNA imaging to visualize the amygdala's activation in response to fear-inducing stimuli. They found that within individuals with higher oxytocin activation, coupling of the amygdala to brain regions associated with fear manifestation were reduced. This connection gave rise to a therapeutic window of opportunity that could be utilized in treating disorders with deficits of the amygdala. Some of these are listed in the figure to the right. Additionally, it is believed that oxytocin influences more intricate neurophysiological processes like anxiety and depression. Anxiety and peripheral oxytocin levels have been linked to generalized anxiety disorder, and additional research has shown that oxytocin may have antidepressant-like properties. These antidepressant-like effects are modulated through oxytocin's activation of the mitogen-activated protein kinase cascade, and therefore the consequential initiation of brain-derived neurotrophic factor expression that triggers neurogenesis and increased neuroplasticity. All of the social behaviors that have been covered are inclusive and form reward positive relationships with other members of their species. However, oxytocin neurons are also hypothesized to contribute to aggressiveness, territory establishment, and other social behaviors that do not support group living. These non-affiliative behaviors are pivotal in upholding these social structures present in mammalian species. A good example of hostile behavior is maternal aggression in rats, where oxytocin increases aggression towards intruders while simultaneously inhibiting aggression towards its offspring. This process is regulated by the central nucleus amygdala. Hubler et al. Demonstrated this connection and found that, like similar to the neurocircuitry in fear, oxytocin modulates the excitatory inputs here. To conclude, I will briefly go over what was covered in this resource. Oxytocin is thought to contribute to a wide range of aspects within mammalian societies. Oxytocin is a pivotal hormone in parental care, and the reproductive mechanisms it controls are responsible for efficient nurturing and raising of newborn mammals. Oxytocin neurons are also important in determining the type of mating system present within mammals and are responsible for species-specific types of sociality. Studies on lemurs and haplorhine monkeys validated these evolutionary distinct oxytocin-determined mechanisms for monogamy and pair bonding. Oxytocin neurons also play a role on non-reproductive behaviors based on same-sex social relationships. Research into similar monogamous and non-monogamous species identified that oxytocin receptor distribution may be responsible for these social behavior differences observed. Due to their more complex social groups and higher order brain functions, primates' non-reproductive social behaviors allowed for a larger spectrum of research. Studies on people revealed additional functions for oxytocin neurons in the control of emotional processing, social anxiety, and trust. Thank you for listening. If you could now fill out the survey on the final right tab at the top of the screen, that would be greatly appreciated.